Yeah, I'm getting
All right, here we go.
So here's a question about I did read through it, you did tell me a little bit, but will you like do another like demonstration of it? Because like I'm a visual doer person.
Yeah, no worries. Yeah, I know that it talks about it in the lab manual. But we're gonna go over like the procedure and the content and everything and what I expect you to know basically.
And did you
No, I will provide it.
If I like downloaded it, I didn't.
Yeah, no, you're good. All right, so we're gonna go ahead and get started. If at any time you guys come in after we've started the Kahoot and you want to join in, it keeps that pin number on the bottom of the screen. So you can always join in the middle if you want to. Okay, we're gonna get going. All right, remember chapter 2 is about chemistry review. Right, which type of chemical bonds involve sharing electrons? Yeah, so it's going to be covalent. If you ever are not sure which one is the sharing, which one is the donating, because there's really only two that we learned about that have some sort of electron moving between atoms. And so just think of like you're codependent, you're in a couple, whatever. That's the one that they're going to be sharing. Okay, that's the idea. All right, hydrogen bonds. What applies to them? Sorry. That was really quiet. No, that's super loud. All right, so hydrogen bonds. These guys are going to hold water molecules to one another. That's because of the polarity that we have formed in the water molecules. Remember, I had told, I was talking to you guys, I had an example, and I can't remember what it was that I gave for you guys, but I came up with a better one for some of the other sections because you guys always get it first and then it just gets better from there. So after I get like into that lecture, I think of some better examples sometimes. My example that I gave the later classes was like if I was playing Barbie with my sister, and so we go outside, my mom tells us to go play outside with the Barbie, and we're sharing and we're being good and whatever. That's just a typical covalent bond, right? So if the Barbie's the electron. However, if I hog the Barbie and I have more time with the Barbie, that's gonna, because it's an electron, make me more negative, but we're still sharing. Okay, so that's where the polarity comes from. And so if I am more negative, that means she has to be more positive because that's what the electron's going to be doing, right? If it's not there anymore. So that's where the polarity of like water comes from, because the oxygen is hogging the Barbie, right? And then if you get just too overzealous with that Barbie, if I were to just take it and run off and not play with my sister anymore, that's an ionic bond, because I'm just like basically stealing it. We could say she's donating it, but we know what my mom would say. She would not say she was donating it. So you steal it and run off. And we do see that with water, right? When we have H2O, it can also exist as H plus and OH negative, right? And when those two things, when you have water, literally just pure water, and you have that shift going on between H2O and H plus with the hydroxyl OH negative, right? Still H2O, they're just, now we have the proton separated off. And they should be equal in amounts, right? Because if you just have H2O and that's it, you would have equal amounts of H plus and OH negative, okay? That's pH. That's what pH is talking about. If you're looking at pH, you're looking at the concentration of the hydrogen ions in there. And if water is where it's equal, then we're talking about if we're adding more protons or taking away protons in that, and you're shifting that balance one way or the other. So that's what that was about. Anyways, the reason I'm bringing that up is because hydrogen bonds depend on polarity in order to exist. It has to be a hydrogen, has to be. And the hydrogen is only ever going to be having a positive polarity in this. So the reason it has that polarity is because usually the oxygen that it's attached to is hogging its electron. So that's when we can start associating with negatively charged or polar things that aren't part of its molecule, right? All right, cool. Just wanted to go over that one more time because I feel like the more we can understand it, the more we are understanding basically, because I'm going to talk about a lot of this stuff as we go through the thing. All right, so what element is in all organic molecules? All right, so it's carbon. That one's a pretty easy one. Remember, you can't just have only carbon. You have to also have hydrogen in order to be considered organic. All right, true or false? Water is a poor solvent. I'm sure y'all are going to do good on this one. All right, excellent. So water is actually a very good solvent, not poor solvent, because of its polarity. All right, what is water's pH? Luckily, I just said this one, so see if you're paying attention. Yeah, good job, guys. What range on the pH scale is an acid? And the fast music is stressful. All right, good job. Most of you guys get this one. 0 to 6.9 is exactly right. Less than 7 is going to be acid. Greater than 7 is a base or alkaline. Same thing. All right, lipids. Tell me about lipids. All right, good job. This is exactly right. It is all of the above. When we're talking about lipids being used to make energy for the cell, we're talking about breaking down fat and then using that to make ATP. That's what we're talking about. We'll talk more about that in chapter 10. All right, which of the following organic molecules is amphipathic? There's one in particular that we talked about. All right, so still majority did get it correct. This is phospholipid. It's that phosphate group, and the lipids are always going to be hydrophobic, water-fearing. Phosphate group is heavy charge on that one. It has friggin' 4 oxygens around it. Of course, it's going to be negatively charged. All right, what is a monomer of a nucleic acid? Talking about DNA, RNA, what is the thing that makes it up? The pieces. All right, nucleotide is exactly right. So that is talking about, we're talking about the phosphate group with the 5-carbon sugar, could be ribose, could be deoxyribose, with that nitrogenous base. That makes it ATCG. So remember that that's what that is comprised of. I'm going to shove that down your throat more heavily in chapter, what is it, 6, when we get to genetics. All right, alpha helixes, I don't like that, man. Helices and beta pleated sheets are examples of which type of protein structure? Where do we see that? All right, great job. That is the secondary level. Remember, primary is like if you had your protein, your polypeptide, that's just a whole bunch of amino acids just stretched out and you just were like counting them off or whatever, naming them off. Think of it as like a long ribbon. And remember, some of those guys are charged. Some of them are hydrophobic. Some of them have kind of weird stuff going on, okay? So depending on who you're right next to, you might want to be closer to your neighbor or farther away from your neighbor. Those associations immediately next to you are what form our alpha helices and beta pleated sheets. And then from there, you can have larger associations with guys way across the way to make your whole overall total crumpled up ribbon, or I think we had said aluminum foil or whatever. And that's your tertiary structure. But you might have to have more than one unit come together to make it a functional enzyme. That's the quaternary structure, right? Just to remind you guys, because that's one that a lot of people have some difficulty with. All right, let's do our podium. And we'll get into chapter 4. Chapter 4 really is some of the first really new stuff that we're going to be seeing. All right, good job, guys. Bask in your glory for just a second, and then I'm going to close this. Good job. All right, here we go. All right, so we're going to be talking about bacteria today. And it's going to be pretty key to understand and at least recognize the differences between these bacteria and archaea, the eukaryotes, compared to the eukaryotes, okay? So I had said this in the review, but I'm going to draw out one more time in case you haven't watched the review or you think you don't need to watch the review, which congratulations. But basically, have I told you guys that if I leave something like this up on the board that has basically answers to questions and you guys get started on the test already, it's fair game, okay? Yeah. That's my responsibility, so if I mess it up, I have to eat it, right? All right. Okay, so if we're talking about cells, things that are made of cells, we were talking about all sorts of microorganisms, right? So we'll just say micro. They can be made of cells or not. So if they are not, we call it acellular or non-cellular, right? Same thing. Please understand that. Don't get confused by it. You're not looking at different terms. Don't overthink it. All right. So, and then we can have the ones that actually have cells. What is a cell? A cell is basically a structure contained within a membrane of some kind that has everything that it needs in it to survive and to reproduce. That's really the definition of what a cell is, and nothing beyond that. So it has to have some sort of genetic material too, but that wouldn't happen. It wouldn't be, reproducing without that. So the given. But yeah, that's the basic concept of what a cell is. If it is contained within a total membrane and is able to survive and divide and reproduce on its own without having to always rely on machinery or whatever from another cell to do its work. Okay, that's what a cell would be. So the acellular guys, we only have two of these. A virus and a prion or prion. I'm not going to judge you guys for mispronouncing words in this class. I'm way more concerned about the understanding the concepts. All right, so cool. So we have that, and then we have the cells. And you can divide this into, let's just do eukaryotes first. These guys have a nucleus. Into the eukaryotes. No nucleus or lack a nucleus, whatever you want to say. I feel like this is something, like, it is basic, I know, but like it's something that some people like to see, just so they can organize all this in their mind. Seeing it out in like a chart like this has helped. And if you guys are of the mindset that like when you study you have to just use flashcards or only copy your notes, remember there's other things you can do, things like this. So use whatever tools you can in your toolbox if you think would be helping you whenever you're studying. Okay, so our eukaryotes have a nucleus, and we're not getting into that chapter yet, so I'm not going to go any further on this. Okay, our eukaryotes are no nucleus. There's two categories that go in here. I should have got that green marker. Here we go. The one I stole. Here we go. All right, so then we have the archaea and the bacteria. I want to be very clear with this. Archaea are not a kind of bacteria. They're completely different things. If you were just look at them face value under a microscope, they look very similar, but they are completely different. I mean, they have like inorganic compounds that make up their freaking membrane and stuff. Like they're weird. So they're not the same thing. And we're not going to talk a lot about them, but all right, so let's go on. So now that we've like kind of cleared that up in our head, we have already set up to understand the differences between the eukaryotes versus the bacteria and archaea, right? And those are talking about the domains there. What else? So if we say that something has no nucleus, we are also saying it has no membrane-bound structures on the inside at all. Okay, because that would be number one would be the nucleus. So we don't have mitochondria, we don't have Golgi apparatus, we don't have the endoplasmic reticulum, we don't have any of that. We just have stuff chilling out in the milieu, in the cytoplasm, right? That's the word for it. So the goo that everything lives in your cell is called cytoplasm. It is water-based and it has some electrolytes and stuff in it to keep everything nice and happy and stable. These are all of the parts in a bacterial cell. I'm not going to go over them right here. We're going to go over them through the slides, but this is a good one to come back to test your knowledge of what they could do and stuff like that. All right. We already know if they're cells, they have to be able to reproduce. They have metabolism. They have to have nutrients, because if you're going to reproduce, you need energy. And what do you need energy for? Like making things, building things, and that's metabolism. Bacteria can also act as a group. colonies, biofilms, nanowires. We talked about biofilms already with like plaque on your teeth and that sort of stuff. Colonies, we've talked about that. It's a single cell growing up into one big pile. And we'll talk about that in lab as well. The sizes and shapes, it's kind of important to understand these. There's really two that are in this class that are actually, I'm gonna totally actually really test you on because I only have two kinds of cells in the lab. So there are two shapes, but you do need to know these terms. And here they only have three terms, not too bad. They have coccus. Anytime you hear coccus, like staphylococcus, right? That they are spheres. Generally, just think of circles. Okay, I don't want to get too, it has all these other things that it could be. No, just they're circles. Bacillus, those guys are rods. I don't know. I always think of bacillus, it's like a bat. I don't know why, but that to me is more rod-like. I don't know how you want to deal with coccus. That's on you, man. But yeah, so that's what we're talking about with the actual shapes of the cell, and then curved. Any sort of spiral shape to it at all, we'd say it falls in the curved category. And that's really the ones that you need to worry about. There's some cells that can have a shape called pleomorphism. That means that if you were to look at them in the microscope, the cells would have all kinds of different shapes. They're just not holding it as well. If you have a cell wall, you're generally going to be holding steady and be nice and sturdy. If you do not have a cell wall as a bacterium, then you are a little bit more flimsy. And so you'll be more pleomorphic. Can't tell what shape it's going to be in. So here's some pictures of shapes and arrangements. At the top, we have the coccus. The middle is the rod, and then the curved next to it, just to give examples. If I am going to be giving you examples on a test, I would know the word, what they mean, and or I'm not going to give you something that's kind of questionable shape. Okay, it's either going to be a circle or a rod, and I won't give you something that's in between. We have some bacteria that like to live in the in-between, but I won't give you those on the test, okay? So rest assured that won't be happening. But you can see this picture of Staphylococcus aureus in that purple, all those little grape-like clusters, right? Staphylo means grape-like or irregular clusters, all right? So staphylo, that's what that means. Strepto, you can see in the bottom, the little chains of the purple guys. Streptococcus is already telling you it's a coccus, it's round cells. And they're in a chain. That's what strepto means. And the way I remember the difference between strepto and staphylo is I think strepto sounds like strip. So it's like a strip of guys. Whatever works for you though. And then there's other ones whose names aren't going to give away their shape at all. Of course, like Legionella or Escherichia. Does Escherichia tell you anything? Is it rod shaped or is it, how would we know, right? But sometimes we get lucky with things like Staphylococcus. It's not only telling you that it is a circle-shaped cell, a sphere-shaped cell, but also that it's arranged in irregular bunches or grape-like clusters. Same thing. Okay, so that's what those terms are for. We'll learn more about those in the lab when we get to that part of the lab. All right, Diplococca. So here's the arrangements kind of spelled out for us. That's nice. I was just going by the pictures. Diplo, probably most of us know what di means too. And it is very clear when they are diplo. And you'll see that sometimes when you look at your bacteria, you'll have a couple of different arrangements going on. That's okay. That's fine. Some of them tend to have a specific kind, like Streptococcus. If you guys have strep throat, it's Streptococcus pyogenes, like 99% of the time. So Streptococcus, then it's going to be the chain of the little circles, and that's what that would mean. So it's pretty predictable what you would see when you look at a microscope for those. Some guys don't have their shape or anything in their name. So if we're talking about Neisseria meningitis or Neisseria gonorrhea, well, I'm sure you can guess what they cause. So Neisseria meningitis causes the worst kind of meningitis you can have. It's the one with the rash. They call it the meningococcus. So I'm telling you there, what they call it, the meningococcus, what shape are those cells going to be? Round, right? Circles, yes. But they also are characteristic and very well known for being diplococci. So it's like one of those things you get somebody CSF, you look at it and a gram stain under there, it'll be gram negative and you'll see diplococci and you immediately know what it is. Of course, you should already be, with meningitis, it's so serious, they already administer antibiotics to treat the worst kinds that they know of all at once. They'll just start it right away because it can be so severely deadly. And then when they get that answer back, then they're going to switch to whatever's preferred for that kind of meningitis. They don't want to wait for the gram stain even. So some things you can almost recognize by that whenever you get to know they have these defining features and whatever. And I told you guys about Staphylo, grape-like clusters or irregular clusters, same deal. And then strepto in a chain. Sarcina is a cubical, that's what it says, packet. It's like if you had it's a cube. It's 4 on each side, right? And that's what it would look like. And they do look like that. You will never see that in the lab, unfortunately, because it sounds like it'd be cool. I've never seen one in real life. So bacilli, so that was, we were talking about the cocci, right? The bacilli can also do diplo, so they can be in pairs. You can also have strepto, so it'll be in long chains of the long rods. And then we also have something called palisades. For the bacilli, the palisades are whenever you have the rods kind of up next to each other like fence posts. Okay, and it's when they kind of divide off of the ends of one another and they just kind of live like that, say like that. All right, that's all I care about for those. Biofilms, we kind of already talked about this, but it's just a whole bunch of bacteria living all together, and it doesn't have to be just bacteria. It could be bacteria and fungi and or archaea and or, you know, protozoa or protista or whatever. So anything can be involved in the biofilm. When we have a biofilm, and it's going to be layers and layers, even in plaque on your teeth, or even if we're talking about biofilms that would be forming anywhere else, one of the most serious concerns in medicine, of course, is biofilm on like a hip, that somebody's had a hip replacement. And most of the time, if you're having an issue with that healing and it is infected, it's from a biofilm that's formed on the joint. Okay, so that's why you take antibiotics beforehand and during the healing process. And they are special antibiotics to deal with biofilms because there's so many different kinds of bacteria that could be involved in the biofilms. They can talk to each other in there. So as long as everybody's happy and they've got food and they're giving off all the sorts of like waste products and signals that everything's going good, everybody knows it's going good. But then if things start going bad in one area, that sort of sends signals out that things are going bad. And at any point they can sort of like decide they don't want to be part of this anymore and leave. So they call that quorum sensing, like a quorum. If you're holding a quorum sensing. We'll get a little bit more detail of that a little bit later, but yeah, you can imagine how it would start. I'd have to explain that to you. Bacteria come and they stick to a surface and the things keep sticking. Congratulations. All right, so appendages. We have things that can be sticking off the edge of the cell for the bacteria. There are two major kinds. Well, I say two major kinds, two major groups of appendages. The ones that help it move and the ones that don't. The ones that help it move are flagella. In bacteria, only flagella will make them motile. Okay. The other guys that won't help them move, guys, are used for attachment or sending, you know, DNA between one another, like sharing genes, like antibiotic resistance, for example, and all sorts of stuff like that. So this is a flagellum. That's a singular, or the flagella is multiple. And this is kind of what it looks like. Look, if it looks like a motor to you, it does look like that if you do an electron microscope and actually look at it. So this is not going to move in a whip-like motion. You can tell just by looking at the motor that it wouldn't move in a whip-like motion. It's going to rotate. And it either goes clockwise or counterclockwise. And that is it. But it's this big old long thing hanging out off the back of the bacterium. And it will be spinning it in order to move. You can imagine that that's not a very specific direction that you would be going, right? If you just kind of be going, how do you control turning and stuff with stuff like that? You can't control the length of the flagellum. That's not like something you can do, okay? So it's kind of just out there. And we'll have to talk about how that works. But first, let's talk about the arrangements. And that actually helps us kind of understand a little bit how they work. All right, we have polar arrangement, which would be monotrichous, would be a single flagellum at one end, mono. Trichous, like is like tricho is hair, so it's hair-like. Monotrichous, one at one end. Lophotrichous, tufts at one or both ends. Amphitrichous, both, right? That's what we know that means. Amphitrichous, one flagellum at each end. And then peritrichous all over the surface. So that one is not a polar arrangement, but somehow got put under that period. So yeah, peritrichous is all over. I don't know how to tell you how to remember the lopho, but that sounds, to me, lopho sounds like a wolf And that has tufts on it. You can think about it that way, I guess. That's what works for me. Amphi is both. Peri, I don't know what. All right. So bacteria using their flagella will be moving, and they have to be moving because of something, right? They're not just moving willy-nilly because that's a waste of energy to just move for no reason. So they're either going to be moving from chemotaxis or from phototaxis. If they're moving from chemotaxis, they're moving because they want to get closer to or away from some chemical signal. Imagine if I had fresh baked cookies at the front of, up here. Everybody would want to move towards that, right? You can smell it. That's your chemical signal that you want to come towards the food. If I put a pile of dog poop up here, you would smell that and want to go the other way. That is chemotaxis. So that's what they are doing in their own world and with their own things. Phototaxis is the same thing. If you're a photosynthetic bacteria, that might be something that you want to pay attention to. Here's how they move. So even though we only spin our flagella in one direction or another, how we move is in runs and tumbles. So a run, we're just spinning our flagellum in one direction, spin, 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 and spin. And then a tumble, we would just like suddenly the other way. And that causes it literally to look like it's tumbling. And then it can keep going that other way or going the same direction, or it can like shift and keep trying to turn around and do whatever it needs to do to get hopefully closer to the thing that wants to get closer to, or farther away from the thing it wants to get away from. And that's really how they go. They can't direct their movement any more than that. But you can imagine that if somebody has just one flagellum, then they can only have that one flagellum doing the work for them. But if the guy has a whole bunch on the outside, you've got more that you can kind of use. You can like spin some of them and not others and whatever to control your movement. And they can do that actually. So it's pretty useful there. So here's a picture of what it would look like if your bacteria were moving around. If there's nothing to attract it one direction or another, then they kind of can just move randomly and it will look very random. But if they are attracted to something, then they're going to have a biased movement based on how they're using their runs and their tumbles to get in a general direction, not in a straight line, but generally in that direction. That's the idea. That's the best you get. All right, here's our first questions already. And we already had just that means we're halfway through this, by the way, pretty sure. I don't know how, but we are. All right. First question, Madison H. Are bacteria always single cell? Good job. Wait, what? Hold on. What was the question? Sorry, I'm like, yeah, cool, yeah. Actually, okay, so our bacteria are always single cell. So when we're talking about single cell organisms and multi-cell organisms, and that's really what this is about. And I know how you're answering the question, right? So we would think of that if we say, no, they're not always single cell. They do biofilms and stuff like that. The question really is asking, are they ever multicellular? And that is asking about like, we are multicellular. And so they don't have to be, like they aren't ever that. So even when they're in the biofilms, they're still individual. So really the question, I guess it's a vague question, and that's my fault, but still gonna give you the point, because I understand where you're coming from. Bacteria are always single cell. No, because they can work in biofilms and stuff like that, but they don't have to, right? And that's kind of what the idea is. So they are single cell organisms in the sense that they don't have to be multicellular, if that makes sense. Okay, that's what that's really getting at. But that's, you thought through it and you weren't wrong in your thinking. So, all right, what is the term for round cell shape? Another foxtrot, Angie. Okay, yes. So I like that you tried to guess something that was related to the words we were talking about. Okay, anybody from Foxtrot know the answer to this one? Round cell shape. All right, I'm going to open it to anybody. Anybody know the answer? Okay, I saw yours first, so Fox, yes. Exactly right. What's your name?
Stephanie.
Okay. Stephanie. I'll find yours.
Alpha.
Okay, Alpha. Yeah. So I'll find yours and make sure that you guys point for it. It's right. Stephanie. And you won't get picked again today. Just remind me if I call your name.
Oh, I will.
All right. So that's the deal. All right, guys. What is the term when cells are arranged in irregular grape-like clusters? This is Charlie, this is Elizabeth.
Is it, strepto? Or
So so it's staphylo.
okay.
I'm still gonna give you the point because you're talking about the right words. You know what I mean? Like when you guys are gonna answer my questions and I'm asking you about what's a grouping or whatever, and you're giving me words that are groupings or at least sound like they were grouping words, then typically I'm gonna give you the point for those because that was a correct word in that area. But that is not the correct term and it's staphylo, but yeah, you knew what it would be if it wasn't that. So good job. I'll give you the point. All right, which appendages can bacteria use to move? This is Cynthia. Yeah, that's it. That's all. That's the only one. All right, and what does peritrichous flagellar arrangement describe? Peritrichous, what does it describe, Melissa? Yep, all over. Exactly right. You guys are really good on this for these being terms that are like mostly brand new for you guys. So you should give yourselves a pat on the back for it. All right, you guys can sit down. We're going to move on. Oh, I did another one. Sorry. We don't have to do it. Y'all give it. Let's just move on. I know that make it right. Right. So, all right. So the other appendages, we have the pili and the fimbriae. All right. The pilus or the pili is multiple. The pilus sometimes provides adhesion, but it's not the most best known for that function. We'll talk about what it does, is the best known for in a moment. And then the fimbriae is plural. They provide adhesion and just adhesion, and that's all they do, okay? So we've seen flagella, they're for movement, and that's all they do, and they're the only ones that do it. We've seen the fimbriae, they're for adhesion, and that's the only thing they do, but they are not the only ones that can do it. The pilus can do it as well, but the pilus has another function, not movement. We're gonna get to it. So these are the fimbriae. It looks like little spikes coming off of the bacterial surface that allows, they have basically a sticky kind of proteins that allow it to stick to certain kinds of surfaces. So it says like down from the bottom, for example, Escherichia coli or E. coli uses fimbriae to adhere to epithelial cells. Especially if we are talking about you getting a urinary tract infection, their fimbriae are what allow it to adhere to the inside of your urinary tract. All right, the pili. And if you guys are there sitting like being like, oh man, a urinary tract infection with E. coli, that would be horrible. It's like 85% of all urinary tract infections are E. coli from your own bottom. So, so think about that. That's why we have that front to back rule, right, ladies? So, so pili, these are, again, that's the plural. It says long structures like our tubes. And that's how I always think of them. And this is what I always think of pili as being for. Generally, this is the more important thing that I want you to learn about pili. Okay. We also call them sex pili. And the reason why is they use this for conjugation. Yes, like conjugal visits. That's exactly the type of word we're talking about here. And here, when they're having their conjugal visits, they are using pili to join two cells together, and they can exchange DNA that way. Pretty similar to conjugal visits. But yeah, so that's the sex pili, and that's their most important function. We typically only see true sex pili in gram-negative bacteria because it has to do with their outer membrane, and we'll talk about that in a moment. All right, surface coatings, S layer and glycocalyx. I'm not really ever going to talk about the S layer because I don't know a whole lot about it. But it's like a protective sort of layer. Cool. I'm not going to ask you about it. I will ask you about other glycocalyx ones, the slime layer and the capsule, and specifically the capsule. All right. We have the slime layer. It's got this loose, gooey, sticky around it. The capsule is more tightly bound and denser version. Like you really just packed it all together, and it's still sticky, still very sticky. And they can both be used for adhesion and also very good, like we were seeing for making the biofilms for sticking to things and then building our other things up in the stickiness. These guys are good for that, okay? That's the slime layer and the capsule. But the capsule can do more than just that for us. So here's a picture of kind of how they'd be depicted. And here's actual literal like staining. So we get that negative stain staining the background. We have the cells stained purple. They are that purple little lines that are inside of those white things. And the white thing is just an area that did not get stained because it is that sugary, sticky coating. It's the capsule. So if you have that white halo around you, you have a capsule. And we'll see that when we do the capsule stain as well. You can see the difference too in the growth on the plate. Look at the gooey on the right there versus the one that doesn't have a capsule. All right, so the other thing that capsules can do, these are formed by a lot of pathogenic bacteria. They help protect against phagocytosis. This is a word that's going to come up constantly in this class, all right? So phago means eat. Cyto or cytosis means the cell is doing the eating, okay? So cell eating. So don't forget that because we're going to have it come up a lot. This is how your immune system works, essentially. You're going to have the base guys like neutrophils and stuff like that coming in and eating up bacteria, which are significantly smaller. Remember, we talked about how eukaryotic cells are much bigger, 10 to 20 times bigger. So they'll come in and just literally like zoop, put their cell around it and then have that fuse, and then they've made a little vesicle with a little bacteria inside it. It's called a phagolysosome. We'll talk about how that works. Again, I know there's a lot of this in this class, but we will talk about how that works later on in the course. So that's phagocytosis, when your like white blood cells are going around eating the bad guys up. So capsules are sticky on the outside, have a sugary sticky on the outside. So it's hard for our cells to grab onto them. And so they can escape very easily from phagocytosis. And I will say most pathogenic bacteria have capsules. I wonder why, right? So it's also used in biofilms. Cool. All right, let's get into the cell envelope. This is where we're going to be talking about the differences in bacteria, where we're talking about gram-positive and gram-negative. If you hear me say it, I will say it like forever, but pretty much from here on out. And I expect you guys to know what I'm talking about, okay? Let's do this. So the cell envelope, this is referring to all of the layers on the outside of the cell, including the thing containing the goo inside, okay? But that entire layer on the outside that's involved in containing and protecting what's inside is called the cell envelope. It is going to always have a cytoplasmic membrane. Cytoplasmic membrane says where it is in the word. Remember, cytoplasm is the goo inside your cell. So this is the membrane containing, it's on the inside most area. So that's your cytoplasmic membrane. That's all we have on our cells is a cytoplasmic membrane. Bacteria can also have a cell wall. In fact, most of them, and that is almost the cardinal rule, that they will have a cell wall. It's very rare when they don't. So cell wall, and what is that? And no worries, we're going to get to that because I'm sure that's going to be super exciting for you guys. Then we have the outer membrane. Some of these guys have an outer membrane. So we will see it as all of them have a cytoplasmic membrane. Generally, all of them have a cell wall. Some of them have another membrane on the outside, the outer membrane, right? And that is not a rare one. We'll talk about why. Okay, so this gets us into the Gram stain. It was made by this guy, Gram. And it's basically going to allow us to see the differences between gram-positive and gram-negative. You are called gram-positive because you stained positive by the stain. And that's why it's literally named it that way. It doesn't mean anything really else. But we have noticed that if you stain positive in the Gram stain, you have these specific features with your cellular envelope. Okay. Gram-positives have thick cell walls, and gram-negatives have a thin cell wall. And because now they don't have that thick cell wall providing all that structure for them, well, they have to have something that helps them out there. So they have the outer membrane. So they're the ones that have the outer membrane. Only the gram-negatives. They're the only ones that need it because they have the thin cell wall. It's not doing much for them. They needed help. So gram-positives don't need the outer membrane. So remember that, because these are big differences here. These are key. And we'll talk a lot about how this disease does this because of the gram-positive outer membrane or gram-positive peptidoglycan, thick wall, or whatever it is. We'll talk stuff like that, like when we're talking about bacteria that grow on your skin. We'll talk about staph and how it can survive that salty environment. It's a gram-positive bacteria. It has a thick cell wall, so it can stay more structurally stable even in the presence of salt. So it has that supporting it. Makes sense, yeah? So we're going to go into some pictures of it. and make more sense of it. This is the example we're given, whatever. This is your, oh my gosh. This is your cell membrane, your cytoplasmic membrane, the inner membrane. So this is inner or cytoplasmic. All right, so that is what that is. So everybody has that. No big surprise there, no difference there, right? We can see quite clearly on this some of the major differences going on here. When I say we have a thick cell wall on our gram-positive, look at that. It's like a freaking tower next to the other guy. That's what we're talking about here. And in fact, it is so thick, the scaffolding, and I'll show you how it is formed. I'm not going to make you guys draw it out, no worries what peptidoglycan is, but how it is formed and stacked up, it looks a lot like a scaffolding. And because it's so tall for the gram-positives, whenever they're dividing and whenever they're, building new cell wall for whatever reason, they have to have helper molecules with them. And those guys are called teichoic and lipoteichoic acid. If you were to get a sample from a patient and sent to the lab, and they gave you the only indicator that they gave is that they saw a presence of teichoic acid, you would know right away this is a gram-positive organism. All right? So that's an identifier for that one. And there is an identifier for the gram-negatives as well. So with the gram-negatives, you can see that little thin layer of peptidoglycan. I mean, it's quite thin. It's that pink layer in there. And then you can see the lipid bilayer, another one on top of it, is literally just another membrane up there. So on the outside of that, they have pores and stuff for things to get back in, so get in and out of the cell. And now we have this whole other layer that things have to travel through, and isn't that nice? And it has some specific kinds of markers on their surface called a lipopolysaccharide, or LPS. LPS, and this is also known as endotoxin. I will spend quite a lot of time in the course trying to hammer into your brain that endotoxin is only associated with gram-negatives and always associated with gram-negatives. Again, you see teichoic acid in a sample, you know it's gram-positive. You see LPS / endotoxin, remember they're the same thing, then you know it's gram-negative. Why do they call it endotoxin? It's endo because it is part of the actual bacterium. It's toxin because it triggers us like a toxin because of severe fevers, inflammation, and all sorts of stuff. And this is what, if you have a whole lot of it, can cause toxic shock response and can be deadly if you have too much of it in your system. So it is toxic to us. Typically, when we have just some of it, typical infection, you're only going to have fever and stuff, but whatever. So that's a toxin. That's why it's called that. All right, so let's talk about peptidoglycan. That cell wall that we saw, especially on the gram-positive, super thick, is made up of primarily peptidoglycan on all bacteria. And most bacteria have a cell wall. Like I said, there's one group that doesn't, but it says a strong but flexible support network. This is what it looks like, right? Here we have, these are sugars here, the G and the M. It's N-acetylglucosamine and N-acetylmuramic acid. Again, I'm not going to quiz you on that, but that's why they have G and M, because that's the difference in letters that are obvious, okay? So those N-acetylglucosamine and N-acetylmuramic acid alternating, and they're linked together, long chains of those. And then across, holding those long chains together, creating our scaffolding, in between, we have what we call a peptide cross-link. So we have some amino acids kind of holding them all together. So that's our peptide. So when we say peptidoglycan, it is telling you what it is in the name. Peptido, we have peptide cross-linking, keeping the things together. What things are we keeping together? Glycan, sugar-based molecules. That's really all you need to know. I'm not going to make you draw it or recognize it or anything. Just be conscious of what the structure is in general. So, yes, so we have the thick cell wall for the gram-positive, many, many layers of the peptidoglycan. So we need help with the teichoic and the lipoteichoic acid. So yes, they help with enlargement during cell division. So that literally means just making more of it when we're making new cells. It has an acidic charge on the cell surface, and it is involved in maintaining cell wall. So if you're having damage or something like that, these guys help you like basically reorient how to put new peptidoglycan in place if you're healing or whatever as a bacterium. So that's what it's talking about. All right, so the gram-negative cell wall, semi-rigid, but not as rigid as what we have with the gram-positives. It's thin, so it's more sensitive, and it's going to be more sensitive to lysis though as well. So we're going to be more flexible. That's what I was trying to say. More flexible and sensitive to lysis. Lysis means breaking open. This is exactly why they wouldn't do well on your skin, and the other guys would. Lysis, it's susceptible, means it's sensitive. No, yeah, I already said sensitivity. Whatever. To like salt causing your cells to rupture, or like too much water when you talk about osmosis, which that's something we'll get into more in chapter 9. But yeah, so they're protected. They're more flexible, but they're not as well protected from those things. Okay. Then, so we talk about those. Those are our gram-positives and our gram-negatives. Those are our bread and butter. And that's the bread and butter, the differences between them. So that's your take home with those, because we will come back to that a lot. Then we have some non-typical cell walls that you will also have to know and understand. We have already introduced the concept of mycobacterium as having mycolic acid wax on its surface. So mycolic acid is a wax. It helps them be more pathogenic. That means cause more disease, makes them resistant to certain chemicals. And so whatever, it's able to hide out in your body and not get attacked as well and not get as cleaned up as well and all of this stuff. But that wax on the surface can make it easier to stain them a specific way. We use something called carbofuscin that we'll learn in lab. It is lipid soluble. So if it's lipid soluble, that means it does well with waxes and stuff, right? So it'll bind with the waxy on the outside, and most other cells aren't going to have that. So it's an easy way to stain only the ones that are going to have the wax on the surface, right? So that's the acid-fast stain in a nutshell. Mycoplasmas, these are the ones that don't have a cell wall. They're the only ones that you need to care about that don't have a cell wall. So they're pleomorphic because they don't have anything to do with a cell wall holding their shape, not even a thin one. They're just cellular membrane. I think they have, yes, I was going to say they have sterols like cholesterol, like we do in our cells, but that's it. So I don't know what else to say. These guys are, why are they important? Why should we care about them? Walking pneumonia is typically caused by this. Cool. Oh gosh, I don't really need to go into too much detail with this stuff, sterols and stuff. I'll mention that archaea have unique branched hydrocarbons rather than fatty acids. I mean, so cool. I'm not going to test you on it. Okay, cool. All right, selective permeability. Luckily, we're not going to get into the chemistry behind this. This is where this concept is going to come back in chapter 9 with osmosis and all that, and hypotonic and hypertonic and stuff that we love so much. So selective permeability. Water and very small uncharged molecules can pass through our lipid bilayers, ours and bacteria. So our lipid bilayers, just the membranes, these guys can pass through kind of freely. If you need to move anything else across, you have to be able to have either a pore that lets it through or an active mechanism to move it through, right? Yeah, especially if you're producing actual toxins or trying to digest things that are outside of the cell so you can absorb them, then that's what we're talking about. You have to do through one of those pores. All right, so this is LPS, and it's a long word, I know. Lipopolysaccharide. It tells you what it is in the name if we take a breath and really break it down. Lipo, it's got fatty acid chain, lipid part. Polysaccharide means more than one sugar. So it's just basically fatty acid chains with some sugar stuff going on. That's really it. I'm not ever going to make you draw again, another thing. Never going to make you draw any of these chemical things. Just know what, if I say LPS, that means lipopolysaccharide and endotoxin. Know that those three things are interchangeable terms.
Thank you.
Sometimes I'll say LPS, sometimes I'll say lipopolysaccharide, and sometimes I'll say endotoxin. They're all the same exact thing. It's just science has met them in different, LPS is just an abbreviation, obviously. But LPS versus endotoxin, endotoxin is talking about how medicine encountered it versus LPS being how science encountered it, right? So that's the only difference. So we have gram stain next. This is going to be basics of how we're going to determine if your organism is gram positive or gram negative. Crystal violet is our first stain. Our primary stain is crystal violet. We'll walk through the steps of it, and it'll make more sense. I feel like if you do it hands-on in the lab, I feel like it makes more sense then. But this is how it works, right? We take crystal violet, which is exactly the color that it sounds like it would be, and think of it as actually being crystals, okay? So if you have a thick peptidoglycan wall, that's all these layers of scaffolding. It's like plinko, or like if you have many layers of like nets and you let balls drop through, right? If you only have one net layer, the balls are all going to be fine. They'll go right through. But if you have many layers, it's going to take them a minute, or they might get stuck in there or whatever, right? Depending on how tall your plinko machine, at some point it might get stuck in one of the corners or something. So that's the idea. It gets stuck in this lattice work, this scaffolding of peptidoglycan. That's what the crystal violet is going to do. For the gram negatives, it won't get stuck there, but it will sit there until you rinse it out, okay? So you can stain things just in general with crystal violet. It's just, you aren't going to know if they're gram positive or gram negative unless you go through this procedure. So you can use it as a simple stain, or you can go through this procedure, and this is the point. So if we were to look at, and it's showing us as we go, but the cell itself, we add crystal violet to the gram positive, it's going to be purple. Of course, it's getting trapped in that peptidoglycan lattice work. Gram negative, you add crystal violet to it, it's still purple, okay, because we haven't like rinsed it off yet, right, with the appropriate thing, and it only is going to come out with alcohol. Next, we're going to add iodine. When we add iodine, grams iodine, it is a mordant, and I just circled right through it, but mordant. And I told you guys, when you hear mordant, think of like mortar, like we have with bricks and mortar that hold the bricks together. So this mordant is going to hold those purple crystals in place like mortar would if they were the bricks, to stick them in place, right? So if you are stuck in there, you will get extra stuck in there in place by the iodine. The iodine isn't really going to matter on the other guy though, because we don't even have other molecules to make them stick together or anything, because they aren't really in there that well. We've still got a purple color because we haven't rinsed out with alcohol, but it's not actually going to make it stick. When you add the iodine, after you've added iodine, there's no color change. This isn't a color change portion, okay? This is like a chemical reaction portion. So this when we add the mordant. But now that we have fixed it in place, we should be able to rinse it with alcohol. So alcohol is our decolorizer. So we add alcohol to these cells. We have locked crystal violet in place with our iodine. It is not going anywhere even when alcohol is there. We did it properly. However, we're finally at the alcohol step with our gram negatives. This is going to rinse out the purple that may have stayed lingering. So here you can see here, we're actually going to literally break down our outer membrane, and that's going to help remove the stain even more. So alcohol just does that. It's one of its properties. All right, so now we're left with a clear cell and a purple cell. Purple cell is going to stay purple. It doesn't matter. You can add as much saffron into it as you want to, because if you don't know what it is, you'll be adding the saffron in, and it'll stay purple. Because that purple is purple, y'all. It is so purple. There's not anything more purple in the world than that. It is dark purple. Then we have the gram negative guy who we rinsed out with the alcohol, who's a clear cell. But in order for us to be able to see him, that's why we're adding the saffron in. Okay, so it's a red dye, so that we can actually see those cells. It's a counter stain. That's the term for that one. It's a counter stain. So now this is where we get to that point in all of this, where I will tell you that gram positives are purple and gram negatives are red. That's how we get there. So that's the gram stain, probably the most important stain we're going to learn. All right, what is cytoplasm? The goo inside the cell. It's mostly water. It also has sugars and amino acids and salts and stuff like that. It's also where the chromatin body, basically the mass of DNA that makes up the genes for the bacteria, are chilling. And everything's just chilling like that. For the chromosomes for the DNA, usually most bacteria are going to have a single circular strand of DNA. That's it. It's a circle. It's weird in our minds, but it's a circle. And they usually only have one, it's big, but that's it. And it will hang out in an area in the cell called the nucleoid, but it's not contained by a nucleus, right? Nucleoid is just a term referring to the area. That's all I want to say, yes. Okay, plasmids, moving on. So we talked about the genome, where that's going to be and what that is doing. Now we have plasmids. Plasmids are also DNA. They're little circles of DNA with like a few genes on them that usually are going to offer protection in some way or toxin production, something like that, but usually like antibiotic resistance and stuff. And basically, if you get a plasmid, which you can get through the conjugation thing, they can like feed one through to you. If you get one, when you divide as a cell, you divide with it. Like you get, like your new daughter cells also have a plasmid. So there's that to think of as well. We use this for a lot for genetic engineering because we can put whatever gene we want to in a plasmid and then put it into the bacteria and then they'll make whatever we want. So pretty useful stuff. That is chapter 8. Okay, ribosomes, whatever with ribosomes, y'all. These are just the things, they're made up mostly of RNA, right? Our RNA, ribosomal RNA, that help make protein. They're the machines that are going to read your messages to make the protein. I don't want to get too much into it right now because it's a lot. So, and endospores, we have, basically these are, I don't want to say this because I don't want to look at endospores. They are, if you are a bacterium, let's say you're a bacterium. If you are a bacterium and you're living your happy little life and everything is good, you don't care about an endospore, okay? But if you are certain kinds of bacteria, 2 genera that I will name, if you are one of those 2 and, you know, things go south, basically, you don't have any more nutrients and or water, something like that, they can make endospores. So it's a structure that they create within themselves that is coated with this heavy protective coating, this cortex, and it is completely dormant. It's literally just holding the DNA and like some ribosomes, and that's it. And so it's just waiting to get woken up. It should be terrifying because I'm telling you, there are bacteria out there that do this and can wait in the soil, and then you come around and step on a nail. Does this sound familiar to anybody? Step on a nail, and all of a sudden you have lockjaw tetanus, right? Tetanus is one that is pretty well known. Creates endospores, and that's why, like, even if nobody has been out there for so long, you can still get tetanus in certain areas because you have these endospores, these dormant guys, just chilling. You go and step on it, and it gets deep enough into your wound. That's why you have to step on a nail or something, okay? They don't like the air, so you have to get them somewhere deep, which is hard to do typically. But let's say you do. Now they're in an environment that they like, so they're going to pop out of their shell. They're not endospores anymore. Now they're going to turn back into vegetative cells, right? So vegetative cells are when they're happy, they have everything they need, all right? Endospores, think of endo, it's called endo because we're making them inside of the cell, but also think of it as like inside, like the hunkered inside, it's resting, it's dormant, and all that, because it's important that you remember the difference between endospores that bacteria do, spores that fungi do, and cysts that protozoa do. Because they're like different, okay? And we'll get to those later. Actually, next chapter. So we'll get to those on Wednesday. So we will do, so that's really it. You're going to cycle between those 2 as needed. You're only going to be an endospore if you need to be an endospore, okay? If things get rough. So that endospore, remember, inert, resting. By inert, I mean completely dormant, not doing anything, just waiting for like water to come wake you up, right? Like dried out. Sporulation means that we are making endospores. All right, these guys can resist heating, drying, freezing, radiation, chemicals. When I say heating, they can resist boiling. This is why if you've ever heard anybody say that boiling doesn't actually sterilize, it's literally these. Okay, you might not think that that's important to you because, like, when are you going to see tetanus? What about C. diff? When y'all going to see C. diff? All the time. And C. diff is one of these, and that's why it's so hard to keep it contained. Okay. Right. So depletion of nutrients, especially carbon and nitrogen, that we absolutely need for us to live. That's usually what's going to trigger it, and it's usually water that wakes them up. So when I decide I'm going to make an endospore within me, then I am the sporangium that is sporulating to make an endospore. So those are the terms that we would use. Sporulation, again, making an endospore, the act of doing it. The sporangium is the cell that is making it. And a cell can only make one, because you're basically turning yourself into it, right? So transformation from vegetative cell to actual endospore is 6 to 8 hours in most species. That should be pretty surprising. Let me surprise you with telling you how long it's going to take for them to come out. This is the typical cycle, but we'll see, and you can actually see in each figure what is going on in the bacteria. It'll duplicate its chromosome, and it'll wall off the duplication and like start making, a covering for it, and then pinch it into a little thing and make more covering. And then once it's got everything ready, it'll just release the endospore out, and so that it can wait for better conditions. And that's pretty much how that will go over 6 hours. So yeah, so it'll just chill. This is our representation of the endospore with its thick outer coating. And that's important for keeping it stable. So germination is whenever we're going to be coming back to life. So think of it as planting a seed, sort of. We're germinating. We're going to start growing back again. So we have our endospore exposed to water and whatever the germination agent is that it was lacking to bring it back to life. This will break down its outer coating and rehydrate it, and then we're going to have a new bacterium grow out of what was inside. Okay, and then once that is initiated, 1.5 hours, y'all. You wake up those guys in 1.5 hours. You step on that nail, and in 1.5 hours, those guys are active and actually multiplying in your body. All right. Here are some diseases that are related to these: Bacillus anthracis. Anthrax. That's why you can find anthrax just out in like fields when it's a cattle-based disease, if you guys didn't know that. But yeah, it'll just be spores and they can be there forever. Clostridium tetani, that's tetanus. Clostridium perfringens, that is gas gangrene, and Clostridium botulinum. A lot of these guys, perfringens and botulinum in particular, these bacteria can be pretty much anywhere at any given time. It's just they take pretty specific growth conditions to activate them. Specific like temperatures and pHs and all sorts of stuff. So it's kind of hard to get botulism unless you are like canning things inappropriately or something. Okay. Okay, this is our questions, our last ones. All right. What is the difference between a gram-positive and a gram-negative bacterial cell? You can completely summarize, that's fine. Kaylin Z.
The difference between the small wall, so like one has this big cell wall.
Yep, that works. All right. Next victim. We have, what is the function of the capsule Eiley seed? Does it, so she's Charlie. Anybody in Charlie want to guess? A point for you guys? I have no idea. I don't remember. You might be.
I don't know, but I think it's it doesn't need a cell wall because it's thick and denser. It's dense.
So I like that you're thinking about it being denser. It but it's not quite right. I think I'm going to go ahead and not do that one. I mean, good try. Good effort. All right, we already have a hand up over here. Yes, what does it do?
Basically, it's just a big, thick wall that keeps white blood cells from moving it.
Yeah, and it's sticky, right? So don't forget that one either.
Is it bad if I check my notes?
I mean, whatever. I'm not, this is not that serious, right?
Check my notes for like 5 seconds and it'll right over.
Okay, so I don't know which group you're involved in.
I'm in up, I think.
All right, we'll find it later. All right, why do mycoplasmas have a pleomorphic shape? And if you aren't sure what pleomorphic is, I'll let you try it first, but if you aren't, I'll give you that hint. Pori M. So pleomorphic, do you need me to tell you what it is?
Yeah. Is it where, it's not where it folds on each other, right? Or is it? Can you tell me what it means?
Yeah, you're fine. A pleomorphic means that it doesn't have a set shape. It can be framed, it's like, it can't hold a shape. So instead of cocci or bacilli, it doesn't have a shape. So why do they have no shape?
Not sure.
Okay, so I will help you through this one. Let's just for a minute, I'll give you a little bit of a hint. What makes gram positives so tough? They're
The thicker.
yeah, that thick cell wall, right? Okay, so if these guys don't have any shape at all, what do you think's going on with them?
Thinner. Or it lacks.
Yes, there it is. Okay. Yeah, so these are the guys that don't have a cell wall. These are the only ones that don't have a cell wall. We got there. Okay. All right, what is an endospore? We have Ingrid.
Dormant.
Yes, that's fine, yes.
Okay.
So it's basically something that bacteria make to go dormant and wait for better conditions. So protective structures, you could say. All right, the next one. Oh, we have more freaking lecture. Okay, you guys can sit down.
54 slides.
I did not realize, why did I have 2 question sets in there? I mean, I must have got confused. I want to look and see where I am in this. Okay, we're pretty close to the end. Okay, that's not so bad. We'll just push through. Okay, so archaea are weird. You don't need to know anything more than that. Some guys make methane as a product. Like we breathe out CO2, they breathe out methane. We kind of already know this when, cows fart or whatever, and they release methane, and we're complaining about that. That is a bacteria in their guts doing that. Halophiles, extreme halophiles. Halo, think of halogens. If you aren't really sure how to call a halogen, we're talking about fluorine, chlorine, bromine, iodine. Those guys are halogens, okay? So halophiles require salt to grow. So sodium chloride, for example. Some can grow in as much as 36%, but they have to basically accommodate it by taking more into their cell. So then they can't handle watery situations at all, like we can. They require the salt to grow because they become so used to the salt. They've adapted to it. Cool. We have the psychrophiles and the hyperthermophiles. Psychrophiles grow at low temperatures, it's freezing temperatures. They'll grow in freezing conditions. And we have the hyperthermophiles. I feel like that sort of says what it is, right? Super high temperatures, so above boiling. So we're talking about 113 degrees Celsius. They flourish at that temperature. 100 degrees Celsius, in case you don't remember, is boiling. Okay, so we can classify bacteria based on their cell walls and cell membranes. This guy, okay. Gracilicutes, firmicutes, tenericutes, mendozicutes, right? These are the terms, kind of general spacing out here. If you even just try to stick to remembering only the gram positives and the gram negatives, then fine, but I'm gonna walk you through how I remember it. Mendozicutes. I had a boyfriend, legit, this is not just a story, okay? I had a boyfriend when I was in middle school, early on. I was 11 or something. And my boyfriend's name was Joey Mendoza. And he was cute. So Joey Mendoza was cute. And that is a long time ago. So that's archaic to me. So to me, Mendoza cutes is archaea. So that's how I remember that, legitimately. And sometimes I have to start here and I can't remember this without going through this whole process every time. If you guys have to do that, that's fine. All right, so the next one that I usually go to is the firmacutes, because that's my next easy one. Firmacutes, they're firm because they have a thick cell wall. Which ones have this thick cell wall? Gram positives, okay? Next, I have teneracutes and gracillicutes. Well, I know that the leftover G in this situation is going to be gram negative. I've just told myself that if there's a G in that situation, we've already taken care of gram positive, so it can't be that one. So gram negative must be the gracillicute. Okay. And these have words that actually mean something, but I don't remember what they are. So teneracutes is going to be the ones that are soft and lack a cell wall. And apparently teneracutes, just think of tener is actually means tender and soft. So tender, just think of it as tender. They're the ones that are soft, they don't have a cell wall. Okay, if you want to figure out what your organism is based on whatever traits it grew on this media, it didn't grow on that media, it looks like this in the microscope, whatever, then you can refer to Bergey's manual. They will use phenotypic traits, which are observable traits, non-genetic traits, to figure things out. And I don't know what else to say. So we have bacterial species that might have subspecies and strains and serotypes and whatever. We don't get in too much of a deal with that. And this isn't for you guys to necessarily stand up for. This is what I used to ask my classes. It was just one question at the end. But what are the differences between bacterial chromosome and a plasmid? And that would be the chromosome is the big actual genetic material of the cell. The actual stuff that codes for that cell's genes is the chromosome. That should make sense. The plasmid is extra DNA. It doesn't need it to survive, but it can help it survive. Things like bacterial antibiotic resistance and stuff like that can be coded on plasmid. That's also circular. It's also DNA. It's just not part of the chromosome. All right, that's it. Let's go up to the lab. Remember to have your closed-toed shoes and your lab coat. Mine is up there already, so. I forgot I had short hair for a second. I saw it in this and I was just like, what is this?